So hello everybody again. We start with the second talk in the afternoon in Spain, mornings in the US, and night in Japan. And now we have the honor of having with us Daniel Fisher, who is a theoretical physicist, of course. He was born in the UK. I didn't know that, but I just learned it checking in the internet, but moved pretty soon to the US. He graduated from Cornell University in mathematics and physics, and then got the PhD from Harvard and got the first jobs at Bell Lab. And then as a professor, he was in Princeton, Harvard, and finally in 2007, he moved as a faculty to Stanford. So all of them excellent, nice places. And now he's a David Starr Jordan Professor of Science in Applied Physics, but also in Biology and Bioengineering. Probably all of you know him for his contributions in condensed matter and statistical physics, including disordered material, spin glasses, superconductors, nanoscale transport, quantum phase transitions, you name it, fracture, I mean, many different things. And for all this work, he was awarded in 2013 with the Onsager Prize. Then more recently, since 2005 or so, his primary research shifted a little bit to biological questions, mainly in evolutionary processes, which is the thing he will be talking today. And he focuses on especially on quantitative aspects, but he keeps collaborations with experimental groups and laboratory on evolution of microbes and also field studies of microbial diversity. And now he's exploring the interplay of microbial evolution and ecology. Finally, let me mention that he's also interested in other biological questions and has done research on the immune system, evolution of cancer, also in systems neuroscience. And today he will be talking, well, you can see the title there, on ecological and evolutionary problems. So please, Daniel.
Well, thank you very much for the kind introduction. The bit you left out is that my mother was born in Madrid. I have more Spanish blood than I do English blood. So I'm going to talk today about some applications of ways of thinking and asking questions from statistical physics on a very puzzling biological phenomena. If I can get my slides to advance. There. Okay, so the question is, why is there so much biological diversity? And this is normally emphasized in terms of the huge number of species, like very different numbers of beetles. Beetles, they do different things. They don't all live in the same place. But recently it's been observed that if you look within single bacterial species, that you can find that in one little sample, that you find a huge amount of diversity. And these are all competing with each other. Why doesn't the fittest wipe the other ones out? I mean, why can this diversity coexist? So the question, the sort of theory coming from statistical physics, one wants to ask, are there sort of large numbers and things that one can take advantage of, and ask whether some simplicity will emerge from the complexity. I don't know how to define complexity, but whatever your definition, biology is certainly complex. So the goal here, somewhat to reverse my title, is really what one should not be surprised by. Is this a phenomenon which one should be very surprised by? Or is it something, if it can appear already in simple models, maybe it's not so surprising? Okay. So here's just an example: Prochlorococcus, enormously abundant photosynthesizing bacteria in the ocean. This is a family tree from one small sample of actually just some subgroup of this. There are many bacteria here from one location, one depth, but three seasons. And the time since the common ancestor has been for these 10,000 years to 1 million years, on the broader group up to, say, 100 million years. So these have been competing all that time. They mix together in the ocean. Over a 100-year timescale, the whole Atlantic mixes in 1,000 years globally. So why aren't some of these better than others? Why don't they wipe the others out? Why is there so much diversity? Okay. So how we would like to characterize it? Well, we could sort of ask, what sort of phases exist, diverse phases, not diverse phases, and so on. What are their statistical properties? Are there some potentially universal features of these? And most crucially, how do we sort of distinguish between different scenarios for explaining the diversity once we have some plausible explanations? Okay. So the simplest observations, and a lot of the data is of this form, is you just take a local snapshot in some region and you look at the abundances or sizes of the population of each of the types in there, each of the strains. Physicists and biologists love Shannon entropy. It's a very terrible measure for reasons I won't get into. But of course, we would like more than that. We would like spatial correlations. We would like to have temporal correlations. We would like to have correlations with the genetic relatedness, with the phylogeny, as I showed on the previous slide. So here's just an example, which is of diversity, not within one species, but of among closely related species. And you look, this is the fraction of the species that have different population sizes. And you notice this is distributed on a logarithmic scale. It's broadly distributed, and the distribution is roughly of the form 1/n. So where on earth can this come from? Well, this is actually a prediction of something called the neutral ecology theory. The neutral ecology theory posits that all the organisms in some group are ecologically equivalent, and everything is given by stochastic birth-death processes and stochastic migration. And there's quite a lot of predictions, but it's very strange. Why on earth should the statistics look as if the species are ecologically equivalent? They're obviously different. One can measure the differences and they respond differently and so on. So if this is something which comes out, and if this sort of holds up these distributions, this should be something that should emerge from underlying complexity interactions, something that looks, at least crudely, roughly neutral. Okay. So we can ask then, is this a general consequence of sufficient biological complexity? Well, we tend to expect that we should always get diversity. So biology is clearly very high dimensional. If I look at all the properties of a cell, or the properties of the protein level and up, the phenotype space, that's very high dimensional. The environmental space, even just in terms of thinking of the number of chemicals that organisms that are floating around interact with and affect them and that they can produce, that's already a very large number. Okay, so what is the basic approach you want to take? Well, the assumption is that the complexities of the biology, the ecology, and most crucially, the evolutionary history, everything's conditional on the past, suggests that the fitness differences, the growth differences between closely related strains, how they depend on subtle changes in the environment, how they couple to other biology, will all be the sums of positive and negative contributions. Why is that? Well, any, for example, mutation, if it were always good, then it would have just already taken over the population. So everything that is going to come in, possible genetic changes, are all going to have sums of positive and negative effects. And so a key caricature then that one would have coming from statistical physics is that this is, sorry, I was trying to get something out of the way. This is, we can approximate these complexities by randomness. And if we do that, we can hope that there's going to be some universality coming from the complexities, the sort of high dimensional character, and the randomness. I'm just trying to get rid of my bar here. How do I do this? Hide floating meeting controls. Okay, there I am. Okay. Okay. So what are the basic questions? So we're going to look at really the simplest possible conditions where everything is mixed together. There are no spatial gradients. There are no temporal variations. So the environment is just fixed. And then we'll ask, can the populations generally sustain extensive diversity even without continual evolution? So if we just bring things together, can they exist in a diverse state without some of them wiping out all the others and driving them extinct? And then the question is, can such diversity generically evolve and persist? Is it stable to evolution? And then the question, which is a little vague, and I'm going to say what I mean, how much complexity is enough to get the extensive diversity? Okay, so the most basic is in terms of one species or possibly two species that are interacting with each other, and then we're interested in the closely related subtypes within a species. Okay, so the interaction between many different species has been looked at for a long time, going back to Bob May. And so N alpha is going to be the number of individuals of strain species alpha. That runs from 1 to K. So we've got diversity of K here that we're assuming. And then the population dynamics is that the growth and net growth death rate of an individual species is going to be have some overall growth rate. Then it's going to have some saturation effect of what's called the carrying capacity. But then there's also interactions with other species that are sort of doing different things, and May started by sort of modeling these as random. So this is the form in which it's usually written, but this already assumes sort of that each species is different from each other, right? The interactions between them we're treating differently than the direct competition with their own species. Okay. This is assuming no birth-death stochasticity, no spatial structure, and in some ways, most importantly, no evolution at this point. Okay. So what are the possible long-time behaviors of such a population? Well, the simplest possibility is that some kind of stable community in which C of the types coexist. That would be a stable fixed point with some of the populations having positive values, the rest of them having zero values. And then the stability has to include the fact that it's uninvadable, that all the other members that are not in the stable community, if you put them in there, they will die out again. They won't come in. So I can have a stable community there. The simplest single community with C equals 3, if I started with 3 species, I could have 2 distinct communities, one with 1 species, the other 2 extinct, or vice versa. Okay, so what about beyond the fixed point? Well, I can have a limit cycle. We know that you can get that in these kind of populations. So I can have a limit cycle that goes around, but I can also have something which is a rather important thing, which is where you get a sort of growing spiral and nothing ever grows extinct. So 2 beats 1, 3 beats 2, and 1 beats 3 here, but you get closer and closer to extinctions. And obviously at some point, the approximation that we don't do discrete individuals and stochasticity breaks down and sometimes they're driven extinct. But if you reintroduce another one, it can come back in again. So that's sort of some kind of heteroclinic orbit. So what we with more than 3 species, you can also get chaos possible. So one of the questions is, can you get stable chaos? Or do you get chaos which is more like this, where the types get closer and closer to extinction? Okay. So those are some of the possibilities. Now, what we're interested in is the population dynamics of very closely related strains. So I'm going to put in a piece here in the way they interact with each other being different than the others. But very quickly, we're going to say, well, there's nothing really special about this. Why is that? Well, there's no particular reason that individuals should compete more with their siblings than with their third cousins. On average, right? They're all doing very similar things. They're all one species. So here I'm keeping the total population size fixed, just from overall resources with a Lagrange multiplier. So breaking off the sort of average growth rates. So this is really the difference between how they grow, just sort of selective differences. If you have a larger S, that means you do better against all others. And then the interactions between them are really sums and differences of the interactions that we had overall, and therefore they're not perfectly reasonable to treat these as effectively random. Okay, and this extra interaction with the competition with the same type, I already mentioned. Okay, so if K is large, which is what we're interested in, what phases of this are possible? What are the possible behaviors? Do we have a large stable community, or do we have some other things? So the statistics of the interactions, what we want to do. Well, we've already said that interaction with the same strain is not especially special, so we'd expect that to be comparable in magnitude to the others. So that's something to keep in mind till the next slide. And then the V's were roughly random, but we of course have to talk about the correlations in them. We can take the mean to be 0, but the crucial thing is the correlations between how type B beta affects strain alpha with how strain alpha affects strain beta. So if they're competing with each other, those are going to be positively correlated. If one's eating the other one, they're going to be negatively correlated. And so in general, I have a symmetry parameter that goes from -1 to 1. Okay. These selective differences, how they differ as far as generalist tendencies, they have to be small in order to get a lot of stability. Of course, if one's better than all the others, it can, by a lot, it can wipe them all out, independent of the interactions between. So how do these kinds of correlations in the V alpha beta arise? The positive correlations, they arise from competition for resources. The antisymmetric correlations can arise from one-on-one competition. If A beats B, then B loses to A, so one's better for the other, and the others, the other way around is worse. Okay, but another way they can arise, and this is a very interesting one, is if I have 2 species, a pathogen and a host, and I have these, and I'll get some kind of block structure where the positive U here is associated with the pathogens benefiting from the hosts, and this negative sign here is the host being hurt by pathogens. And there'll be matrices with not all elements will be the same, so there'll be randomness in the differences of those from their mean. Okay, so this is a particular structure which, in thinking of terms of biological applications, is I think really the most promising direction, is thinking of this host and pathogen, and we'll particularly be interested in bacteria and phages. Phages are the viruses that infect bacteria. Okay, so what is the phase diagram of this as a function of parameters? So the way I've written it, we basically have the symmetry parameter, and then for the time being, we're keeping this parameter, which is the extra interactions of things with themselves. Okay. Well, assuming there's an extra interaction, direct competition with your close relatives, that's more or less, it's like assuming there's a niche. So I could call this a sort of niche type interaction. So there's been progress on this by several groups recently, including particularly these people. Well, if the, I should mention, over here, there's a symmetric V, and then there's Lyapunov function, and things simplify, but they're very non-generic. What happens up here is if I get enough self-interaction, self-competition, then there'll be a unique large stable community with about half or more of the strains coexisting and the rest going extinct. However, for this, you need the interaction to be bigger than the square root of the number of types, bigger than the other interactions by that. And as I said, there's no reason that that should happen. If it does, as we increase the number of strains, we'll drop below this line and we'll go into some region that we don't know what happens. All we know is that it's not a large stable community. Now we're going to be particularly interested with going along this line, where the Q down is at order order 1. The self-interactions are not special, and then we have the symmetry parameter there that we have to consider things as a function of. And it turns out there's a very special point over at this end, very special, pathological in some ways, but turns out to, in some sense, give us the behavior, understanding the behavior of a whole wide regime throughout here. So initially, we're going to concentrate on this point. So we've got negative anti-correlations in the interactions. We're going to focus on that and then try to sort of extend to going into this, this region. Okay, so we have this niche phase, doesn't exist, it's destabilized when we add more strains, which Bob May first knew about, and then we want to ask what happens in this region here. Okay, so as I said, generally self-interact not special, so we've only got the gamma as our parameter. That means generally there's no Lyapunov function. We can't make arguments in terms of energy and so on. Now, if we had symmetric correlations, we can ask questions about small stable communities, whether many of those exist, and the questions about the maximum community size and so on. Can you get different things existing in different places? That's something we're currently exploring, and I'm not going to talk about more here. Okay, antisymmetric correlations. When the correlations are antisymmetric, then there are no stable communities. Okay, if there are no stable communities, what happens? I can obviously get some kind of dynamical state, but that might drive things extinct. So what'll happen at long times? Okay. So there's a special model, which I mentioned. This is the perfectly antisymmetric model, first studied a long time ago. And in this case, there is a unique uninvadable fixed point. Okay, it turns out it's actually marginally stable. Many of the species will go extinct, typically half of them. But the ones that remain are sort of marginal. They have a fixed point, which is where each of them have a fixed point value. And I'm going to call these alpha because they're actually going to parameterize things even when you're actually not at the fixed point. Okay? And the rest of them go extinct and have fixed point value 0 and can't invade. Now, it turns out there's a special sort of energy for this model. So this is basically logarithm of the n alphas. And this acts like a Lyapunov function. Is the derivative of this is negative and transient. But once things have gone extinct, it actually becomes a constant, which of course depends on the history, which ones have gone extinct. I'm sorry, it depends on the starting conditions. And there's a whole family of states associated with what this constant value is. Okay, so for the surviving community, the dynamics of volume preserving in log of the population size, that's sort of natural variable, and the dynamics are stably chaotic. And turn out that the statics of this, the snapshots, look exactly like equilibrium statistical mechanics for individuals with these energies. So each n, this is the energy for them. This is a sum of alphas. So this is just like independent particles, and there'll be some temperature which is associated with what the average value of the energy is. And you see that's going to be associated with the spread in sort of logarithm of n alpha we parameterize by this energy. And so we have a whole family of states as a function of this effective temperature. Okay. What is the steady state distribution? Well, they're just independent particles. We have a constraint maybe on the total number. So you get these power law distributions coming from the exponentials of the logs, and these depend on these parameters which are set by the fixed point values. Amazingly, this full joint distribution of all of these is identical to the prediction from neutral ecology. Neutral ecology just posited stochastic birth-death processes. They're very ineffective in large populations like bacteria. The dynamics is completely different. These are going racing up and down in population, or even on a logarithmic scale, they're racing up and down, as I'll show. So the dynamics are very different. but the statics look exactly the same. So clearly we can't conclude anything about the mechanism underlying it if I see that the abundance distribution here just looks like neutral ecology. This is another mechanism to get that. Okay. Okay, so what we will see, well, if I take large temperature, this large theta, that corresponds to wild swings in log n, and that turns out to be the regime that is instructive as far as what the behavior is more general. Each type is distributed roughly uniformly in log n, but the averages over time, which come in here, they're dominated by the enormously large n, because it's sort of things exponentially distributed, this logarithmic distributed, dominated by the biggest ones, and that plays a very important role. Okay. Now, what about the dynamics? Okay, I've said it's not this stochastic dynamics. This is deterministic dynamics. Things are coming up and going down. Okay. So this special model, this has what dynamics might call kill the winner. It's more generally true with antisymmetric correlations qualitatively. And so if I consider this black line here, so this is the population on a log scale of one of the strains. Well, here, these blue ones are large. Well, those blue ones I've labeled blue because those are the ones that positively interact with the black. So they tend to bring the black one up. When the black one comes up, what does it do? Well, it then produces a negative interaction on the blue ones because the antisymmetry, so it pushes the blue ones down. But it gives a positive interaction on some other ones that's shown in red. So these red ones will then start to come up, driven by the black one. So what do they do? Well, those red ones will then drive back down the black ones again because anticorrelations, this will come back down again. So you get this sort of blooms and bust characteristic where you come up very fast on a log scale, you fluctuate around and you come back down again. Later on, you come back up again, and you get a steady state here where everything is spread out on a broad range. If you look at the average of each one, it's going to be dominated by when it's very large. And it turns out the important part is they all get to be very large, at least some of the time. Okay, so how can one try to understand this this behavior? Well, what one can do is try to do use some of the methods that were developed originally for spin glass and other systems. Is one could try to do a dynamical mean field analysis. So what does one do? One says instead of treating all the interactions between all of them at once, one focuses, say, on one strain here, looks at the population of that, and tries to look at it in the effective interactions coming from all of the others. So it's just like a normal mean field. And of course, I have to do some self-consistently because it's going to affect the others. They're going to affect it. There's some feedback there, and that plays an important role. Okay, so there's an overall part here, which is coming from the average effect on the growth rate or death rate of all the others. Then there's the interaction term, if we still have that. Then there's a noise term, which is coming from all the others. There's a Lagrange multiplier to keep the total population constant. And then there's this piece here, which is the feedback coming from the others. So the feedback is that if I affect you, then you're going to affect back on me as well. Okay, and the sign of that net effect, that feedback, is going to be the sign of the symmetry parameter gamma. And we're particularly in the case when this is gamma is negative, which is sort of looks as if it's going to stabilize things here. But then we have to do it self-consistently. We have to, this R is the response of the other populations to their driving force, and the correlation function here is this, and these have to be determined then self-consistent. So that's the challenge, the theoretical challenge to do that. Okay. So these are the roughly Gaussian and so on, but it's the correlation functions, the response functions that need to be determined. Okay. However, the problem with this is that the normal methods for treating these problems, which is you can write closed equations for the response and correlation functions in this mean field theory, you can't do that here. So one has to do sort of full asymptotics of the behavior with things fluctuating up and down, treating this response function and looking at what happens. Okay. So what are the consequences? This is coming now from the predictions, but I'm showing the results from the from a simulation so you can actually see it. So here we've got plotted on a log scale, and I went to frequency units, which is the fraction of the population in one strain. So this is showing a few strains out of 300 that are surviving there, and you notice that all of them bloom up to high numbers. So they all bloom up to become almost dominant in the population. It turns out anything that doesn't bloom up will go extinct. It may come up for a bit, but then it'll gradually go extinct down here. All the surviving ones will bloom up, and they distribute over this log scale. And there's multiple time scales in here. If you want to be careful about it, it turns out it does somewhat of a random walk in log space. L is a log variable, but with a non-trivial exponent. And the dynamics are not at all like neutral drift. They're also not like randomly fluctuating environment because there's very particular self-consistent determined correlation. So it's like it's in a stochastic environment, but the stochasticity has to be determined self-consistently and is arising from the interactions between all the different strains. Okay. So that's the basic behavior. Okay. What happens if we go beyond this special antisymmetric model? Okay, well, then the fixed point is destabilized. You get sort of heteroclinic chaos, like this example I showed you with 3. The fluctuations get wilder and wilder. They go extinct. You don't get to anything stable. You may, in the end of a stable limit cycle with a few of them non-extinct, but you don't have a diverse state anymore. So this kills it. Okay. Can the chaotic phase somehow be stabilized when you go away from this special point? It's unstable at the special point. Can it be stabilized? Well, there's a long history of thinking about stabilization of diversity coming from migration. Normally what's done is you assume there's a mainland population with many species. They come into an island. These can fluctuate away, go extinct, but then they get repopulated from the mainland. The problem is this assumes that there's a persistent pool in the mainland. So it just replaces the problem of understanding diversity on the island with understanding diversity on the mainland. So we don't want to do that. But we still want to consider spatial structure. So we have many islands here. They're all identical. We have migration between all of these. So it's sort of mean field spatially. And we can get global extinction where the total population of a strain across all the islands will still go to 0, can go extinct. But I have to ask, can we still get many types surviving where they'll spread around the different islands, be chaotic on the different islands and so on? This has been looked at also by Murray and collaborators, and in particular the work with my former students, Atish Agrawala and Michael Pierce. Okay. There's actually another related mechanism, which is sort of a temporal one, is if the strains have some kind of spore where they go into a resting state where they gradually die, but they can regerminate and come back into the population again. Of course, in the long term, if they're not replenished from the active population, then they're going to die out. So I still get extinctions. However, it turns out this longer-lived pool can play a similar role to the effects of the other islands. And in ecology, this is known as Chesson's storage effect. So we want to ask, can this occur? Okay, so what we want to consider then is a Lotka-Volterra model again, but with multiple islands. So we've got large number of islands. You can move from any island to any other island. That's just a migration. So it goes from the total population coming in. They interact only within their own islands. So the J here is the island index. They only interact on the same island, and otherwise things look very simple. Okay, so what will happen? Well, now we can do a spatio-temporal mean field theory. We can sort of replace this by an overall average over all the islands. If it's chaotic, one might guess one can replace that by an average over overall time. Okay, so what happens? Well, this system goes into a very robust spatio-temporally chaotic phase. The crucial bit is that the islands desynchronize, the chaos desynchronizes. So what does that mean? It means if I get a bloom on one island, it can feed the populations on the others, which if they happen to be almost extinct at that point, or even have gone locally extinct on the islands, it can then bring them back and they can bloom up again. So you need those blooms. Each island being roughly independent chaos is what sustains this population. Some of them go globally extinct, but a very large fraction actually will coexist and persist. Again, you get spread out on the log scale, and this purely antisymmetric model on one island is actually a good caricature for what goes on. This sort of how we understand this is building on what we had for that very special model. And that, of course, has got a long tradition in statistical physics of having special models, but they give us the sort of seed of trying to understand the behavior more generally. Okay. And I said the crucial bit is they all have blooms. And the dynamics on each island is roughly like the antisymmetric model, but they don't go completely extinct because they get migration in from the others. Okay. So here's a picture of it. Here's a strain. This is now showing one strain, but across 10 islands. On again, on a log scale, fluctuating up and down. You notice this one then gradually goes extinct. Okay, but then here's another one where you actually just start in one island. If you start at a time when conditions are good, it can bloom upwards. It then goes extinct again on that island, but it's seeded a bunch of other ones. And eventually this goes on and gets into a steady state. And this sort of lower bound here is set by the average migration from all the other islands, sort of floor and below which it won't go once it comes in. And there's one in steady state, and you see all of the strains blooming upwards. I sometimes get little local extinctions, but things get rescued. Okay, so this is a very robust phase. You can put in finite populations. You actually have extinctions. as long as the migration is big enough overall, you still get a substantial fraction of the ones you started with surviving for a very long times. What happens if you have a finite number of islands? Well, then they actually survive for time that's exponentially long in the islands, and this will depend on the strain. So some will go extinct in shortish times, some in medium times, but some will persist for exponentially long times. And in practice, by the time you've got 20 islands, you already get a very large fraction coexisting. Okay, and so the dynamics of this is complicated, and it involves this asymptotics on this dynamical mean field theory. Of course, much more interesting than islands is real spatial structures. They have a string of positions or have uniform behavior in space and spatial dynamics of things moving around as well. We're in the process of trying to understand that now. It appears that, again, the phase is very robust. The nature will change somewhat, but and we believe it should persist with real spatial dynamics. Okay. So what does this say? We've got a phase here, found this robust phase. In a given system, of course, we don't know if it exists, but it is something which, if it exists, then small changes of the system, the interactions, the overall environment shouldn't destabilize it. Okay. But how did we get this? We just took a whole bunch of strains and brought them all together. Nothing evolved. We sort of assumed, okay, they evolved separately, we brought them together. But we really have to ask, can this is a phase that is stable to evolution? Okay, in particular, can it evolve? If we start with it, bringing things together, will evolution destabilize it? Will evolution give us more diversity and so on? Okay. If the evolution is very fast, you're always producing new variants from evolution, then you get some diversity just from that, and we understand that quite well. But that tends not to persist for a long time, and certainly won't persist for the tens of millions of years, hundreds of millions of years. Some of these species have been coexisting and competing with each other. So the hardest one to sort of understand is when the evolution is much slower than the ecology. So we'll try to understand things in that limit. So what happens? Okay. Well, what we can do is we can take initially our strains. So we've got a pool of strains. We've got some starting number of strains. Some of them have gone extinct. So here's our number we start with. It drops down right away. Some of them have gone extinct. And then we just start adding more strains. Okay, so we start adding more strains. These gradually build, can gradually build up. As the strains, we add strains, sometimes a strain will destabilize things. I get small numbers here. If I didn't start with very many to start with, you find you actually get crashes. So I start with a small number, modest number, I can crash down to zero. And then it turns out it's really difficult to get it going again. You only have small numbers of strains. You never build it back up. Okay. But if you start with some sort of intermediate number of order 100, then the number will tend to fluctuate around, eventually grow. And you see here, the number just grows, will grow linearly. Okay, so this looks as if it's really going into a phase that above some critical number, which we don't really understand quite why this is of order 100, it will tend to grow linearly. And once it gets over a certain size, it's extremely unlikely to have a substantial crash. So it looks like not only is this phase stable to evolution, it actually tends to get more and more diverse as you add more and more strains. Okay, so that's really an evolutionary part. But again, that's bringing strains together. What we can ask instead is if we take the existing strains and mutate them. So we can take a strain that mutates, it's then the mutant is now correlated with its parent. Its interactions with others are similar to the parent's ones. They have a correlation, which is, of course, 1 is perfect correlation and 0 is no correlation. So the 0 is like the case over here, where I'm just bringing in new strains. And this is then looking at the function of the correlation. You find unless they're extremely closely correlated, you again get this linear behavior coming up. If they're very closely correlated, you get a lot of replacements. The most likely one is a successful invasion will eliminate its parent, and you'll get both coexisting. But if there's only slightly less of correlations between them, then a lot of times the parent and the mutant can both coexist. And again, you get this behavior coming up. Okay. So what this says, since it can occur in very simple models, is that maybe we shouldn't be surprised by having a highly diverse phase with a lot of very close relatives whose interactions with each other are not very different than their interactions with distant relatives. Anything that tends to make them compete more with their siblings will tend to make this more stable. If you go even further, having them very strongly interact with their siblings and much less with their neighbors, then you can go to this large stable community. But there's a whole wide range where you get this spatio-temporally chaotic phase, and as you evolve it, it goes. Now, this is we see occurring already here, but I did a bit of a cheat. Okay, so it's known that one of the problems with diversity is whether you get generalists. So you can have a lot of species doing somewhat different things, but sometimes you can get a generalist that come in. It doesn't do any given thing as well, but it does everything or a whole many things somewhat better than the others overall. And that can come in and take over. Okay. So what we have to ask is what happens if I have these generalist mutations? What does that mean? I assume that there's some parameter also which determines how well they do against all the others. Okay, so we actually put this in separately. So we're going to the extreme limit where the distribution of the select differences, any mutant can be better overall, as well as being different at how it interacts with all of the others. Okay, but it's still strongly correlated with the parent. So the behavior depends on the tail distribution of what I allow. Basically, if you look at the number of successful invasions, meaning something actually manages to come into the population, so you could observe it, you again find that the number that persist grows still roughly linearly, but with much more noise to it. However, the whole process gets slower and slower. With the particular way we've set up the interactions and things, it gets slower and slower, gradually creeps up into the tail of being better and better overall. So they all gradually better overall, but they're still gaining a lot from the interactions with each other, and it gradually creeps upwards the diversity. Okay. Now, with somewhat different assumptions that end up with sort of sparser interactions and things, you could get that much more like the behavior we had before, where it grows up linearly in time. But we haven't explored that in detail yet, so that's still a more or less a conjecture. Okay. So this is what it looks like on average. If we look at this here, you see that they grow up. If you look at the S's coming into how good they are as generalists, that gradually creeps upwards, and they all become very similar to each other. So this is not surprising. In some sense, if some are just worse overall, you would expect those will have gone extinct. Okay. So even with putting in generalists, we get this phase. It does continue to diversify, but it does it rather slowly. And here you can actually get crashes. So here you've got quite a big number and crash down. Okay. Now our analytic understanding of this is far from complete. So we sort of have conjectures and scaling that really says that this will continue to go up. But that's something which one would like to understand much more of. The problem is all the things which survive, there are the correlations between how they interact with each other just coming from conditioning on the fact that they coexist, and they've coexisted for a long time. Okay, so you can't treat things as being a nice random ensemble anymore. You have to get that ensemble of the interactions conditional upon the whole history of evolution. Okay? And that's a very difficult thing to do, and it's only being able to do it in some particular kinds of models, which I don't have time to talk about today, where you look at things that are much simpler, where you don't follow many species altogether, but you do treat the conditioning on evolution. Okay. So now just the last question is how much complexity is needed to get this high diversity? Okay. So I'm going to treat the interaction in the organisms via, say, the environment or in some kind of direct interaction between them as being sort of much simpler than the overall complexity of the organisms. And some organisms that are pretty simple, that's not unreasonable approximations. And then I want to ask how high a diversity phase can exist, and how will that depend on this dimension of the ways that the organisms interact. Okay. So the traditional view here is to look at what's called community resources. They interact via consumption of D different resources, or more generally D chemicals. In that case, V becomes basically a rank D matrix, and the maximum size stable community is just set by the number of resources. So if you start with many strains, they can't coexist, only a small number can survive. Okay, that's the classical ecological result. Okay. However, we can look at this in this context of the bacteria and phage, where I've got many strains of each. So I have many strains of the phages with population P, many strains of the bacteria with population B. They have an interaction matrix that looks like this. And these will be correlated because, of course, if phage mu is worse for bacteria J, then it'll be better for that phage. And so there are correlations then in here, and because that minus sign there, that give overall antisymmetric like correlations. So now we can say that we've got an interaction via the receptor on the bacteria, which the tail of the phage attaches to. So the interaction will just be the dot product of these d-dimensional vectors. That's the interaction energy. And then we will say, well, the bacterial death rate coming from this infection will be a function of that energy there. And phage population growth will also be a somewhat different function of that energy. Okay. So we can study this problem and ask what happens. Okay. Well, when generally one can get into a spatially temporal chaotic phase, it's very similar to what we had before. If the F and G are linear, then again, you have a rank D matrix and you get the maximum coexisting diversity is small. It's just of order D. Okay. However, if you have sort of saturating diversity, which is much more reasonable biologically anyway, then even with low dimensions, and it turns out D around 5, you can already see this happening, you can get a community of spatio-temporally chaotic community with very many existing, much more than order D. And again, it seems to be going continually up with time as it evolves. There's no specialization of the phage strain. They just sort of gradually chasing around the bacteria, trying to change to get away from the phages, and the phages are trying to catch them. Okay. So this says that even with low complexity, one can get a highly diverse phase. Okay, and to me that was actually one of the surprising things that came out from this. So the conclusions. We found that there exists a robust spatio-temporally chaotic phase with persistent diversity. The best application for this is the host pathogen, but we think such things can also occur when you're interacting via chemicals and you've got things via resources, but that's an open area. This phase can not only exist, but it can evolve and continue to diversify. And what does this tell us? Maybe that extensive coexisting diversity, fine scale diversity within a species, is maybe not so surprising. It's not an explanation for it at this point, but it's at least a scenario which actually has some predictions. If we have this bacteriophage system, you don't actually need much complexity to get this. Whether you can get interaction with chemicals, as I said, we're not sure yet. The effects of real spatial structure I already mentioned, and that's certainly an important part. One of the nice things of species in the ocean is they all get mixed together overall, and so we have some sense of what the spatial mixing properties are. For most bacterial species, we don't have much idea of that. Another possible sort of application is bacteria in the gut, which can get from one person to another. So that's like a bunch of islands, but your islands are moving around, so you sort of get spatial, feel like interactions. A crucial question, though, is what other possible highly diverse phases are. Other phases where they will can coexist, say, in different places in space, where you have different subcommunities in different places. They can not invade each other fully, but they can, of course, mix a little bit and come in a transient state. Perhaps one can get a highly diverse phase like that. And there's some guesses that maybe that's what's going on with guts, where you've got a few important species and strains within different people that are very different from each other. They can invade, you get new ones in, you lose old ones, and maybe you can get highly diverse in the overall community across all people's guts coming from that. Of course, what one would really like to do is talk about the connections with reality. One would like to have some predictions, or at least postdictions. This mechanism naturally gives you very broad distributions of abundances, say like the neutral ecology. Very broad distributions of abundances are also usually seen with species and in the little limited data that there is also with strains within a species. But I would certainly not want to claim at this point that we have an explanation for it. Okay, so the crucial thing to sort of go away from this, one is that statistical physics methods are very useful for thinking about these problems. But the other one is this sort of role of theory in saying what's not surprising. Right? There's a history of this with neural networks and things, of finding these ways with random interactions that you can store information and things. That's not exactly how it's done, of course, but it tells you, well, okay, that's a sort of toy model that shows us that something can occur without invoking all kinds of special features. And the other crucial bit I say is that this can evolve. It can exist conditioning upon past evolution, not just starting it off from scratch. Okay, thank you very much, and I hope there's some questions.
Thank you very, very much, Daniel. I personally enjoyed a lot this talk and your chaotic face. And we have Yuhai too, who has a question. So please unmute Yuhai so he can talk. Yuhai, hello.
Oh, hi. Dan, thank you very much for a nice talk. So I have one small comment and a question. So starting with a small comment, I actually it's agreement with you that something that is actually seemingly surprising is not that surprising. I'll give you the context. The fact that you have competing species and yet no one is winning was shown in the previous work that I'm not sure you know this work I did with microcross in the context of rotating convection, where you have.
Yes, I do. I think there's a reference to it in our paper. So this is with a small number of species and getting into a spatially temporal. Yes.
Yeah. The model, of course, was developed for a different physical system, right, or in rotating convection. But the motivation was actually really part of the motivation was actually Robert May's model about three competing species, the rock paper model, where they see that there's a puzzle there that you go into the clinic orbit that eventually everything distinguishes except for one. Now that was solved by the inclusion of the diffusion term, which basically is your transport term that captured by your spatial. So that's a simple comment, not in disagreeing with you, but in agree with you that something seemingly surprising is actually not that surprising.
Right. Yeah. So that's really this case where you have three species with here one beating two beating three, and with spatial structure this gets this coexists. So that's certainly I know about that very nice work. In fact, I remember at the time when you guys were doing that, and then vaguely remembered and went back and found it. So I think that the crucial question there is this question, what happens if you add many, right? And the thing which I'm making the argument is, in fact, stabilizing when you have very many of them, and stabilizing very many of them is sort of the generic.
I agree. I'm not saying that. Okay, yeah. So I agree with you that this is a very nice work that you have done. Yes. But I do have a question though, related to your actual model with many interacting species. So actually, I have two questions. One is that is there sort of evidence for the anti-correlation of interaction? Maybe you answer sort of that question at the end, saying that this may be the result of evolution itself. That you introduce a species that.
So I think as soon as you get the two species with host pathogen like interactions between them, then I mean you have to have that anti-correlation, right? Because if one phage is better at killing a bacteria, it's worse for the bacteria. Whether you can get it with interactions via the resources, the simplest way is if you sort of have some reasonably local competition, then everyone mixes, then you have local competition, then everyone mixes again. And so you still have a sort of mixed spatial structure. The difficulty there, I think, is the instability probably to getting sort of generalists, maybe more unstable there. We don't know yet whether if you have the resources sort of being replenished at a rate and eaten with space structure there, whether you can get into a similar phase. The models that people have looked at look similar enough that I think this might be possible. So you don't need literally to have the antisymmetric correlation. That's what we have in the models here. It turns out if you just take independent interactions, and this is Roy and company looked at this, then if you assume that there's some amount of niche interactions, but not enough to give you a stable community, then you also go into this chaotic phase. And it certainly will persist somewhat towards the positive interactions as well. If you go all the way to strongly correlated interactions from competition, but not perfectly, so there's not a Lyapunov function, then it seems, and this is again Roy and company, that what you get is you get a temporary state which is almost stable, but everything else is down. And then one of the ones that's way down at low population found, geez, conditions are good now, I'm going to come up. It destabilizes this, some other one goes down, then you have a long transient where these are stable, then another one comes up and so on. And you can get that if you don't allow complete extinctions. There's a steady state that looks like that. So again, it looks chaotic, but there's sort of the separation of the timescales of what's going on. So I think that's a very interesting possibility, and also hoping people will explore that
Agreed. Lastly, that one technical question, which is that I guess you assume these interaction matrix are full.
Right.
They should be. I mean, have you considered a sparse limit?
Yes. Yeah. So I mean, as you know, as with most things with large matrices and stuff, if it's sparse but there is still quite a large number of entries per row, then the behavior should be pretty similar. There is a question of whether when you evolve, even if you start with it being non-sparse, do you tend to produce enough sort of hierarchical structure coming from the genetic relatedness that you will end up looking more like a sparse matrix with bigger variabilities and so on the effects? And that we don't know yet. That I think is an open question. There's you can do models which evolve in a sort of natural way, but where the interactions are still relatively sparse. Of course, one knows that if the interactions are if things only interact weakly with ones that are far away phenotypically, that you can of course get lots of niches, but it's not clear how stable that is. So even in the sort of traditional things where you get many niches by all doing different things, how stable it is under evolution is, I think, still pretty open problem. Yeah, those are all important extensions, interesting extensions to me. By definition, are important.
Perfect. So thank you. Hi, there is another question from Tobias Galla. Tobias, you can talk.
So can you hear me? Hello.
Yes.
Hi.
Yes.
Yeah. So thanks for that great talk. I enjoyed that a lot. I have also a comment and a question. And my first comment is regarding the model on where you have different islands or patches.
Right. Right.
So we've also looked at these in the context of sort of Turing pattern formation. And what we found is one key element that can change the behavior quite drastically is whether the interactions in the different patches are the same or whether there's variation. So whether you change the matrix from patch to patch.
Right. Right.
So I don't know whether you looked into this. I just
no, but let's see. Guy Boon and collaborators have looked at that, where they in the model where they don't have this, where they have sort of independent interactions and some niche term as well, and they find that the variability between the islands tends to make it more stable.
Yeah. So it depends exactly. So there's work by Gravel et al. And then but it depends whether you have trophic structure or not also. So you have this model where you have just gamma equal -1,
right.
But each species, there's no systematic distinction between predators and prey, right? So
I can do, so there isn't in the model I wrote down, by the time I go to the bacteriophage model, I have this block diagonal structure. Correct. The dynamical mean field theory is essentially identical. And so there I really do have the predator and prey.
But these two things are very different, because in the case where you have two distinct groups of species,
right,
you have like an inhibiting group and a kind of enhancing group.
Yeah, it turns out it isn't very different.
For example, you can get Turing patterns this way, we found. If you have two systematic groups, but if you have one group that's all mixed together, you don't, right? So but
the model that I talked about at the end with the bacteria is exactly two competing groups.
Exactly.
Yeah, and it doesn't. But there's no overall patterns in the sense of there's no tendency overall for the phage population to bloom and the bacterial population to go down. The overall dynamics is suppressed by having the many different strains. Each individual strain will do these blooms and bust. But if you look at just measuring the total number of bacteria in that species and total number of phage in that species, that won't have very much dynamical structure to it. There's regimes, I'm sure, and I think maybe this is what you're saying, there's regimes, I'm sure, where you also get the sort of collective dynamics as well of the whole group of the bacteria and phage coming up and down and forming interesting spatial structure, and that would be...
I think that's fine. So then, I don't know if there's other questions, I'll let other people ask, but I had one question.
Go ahead.
So that was about the model where you start with a given number of species and then you allow new species to arrive.
Yes.
You said somehow that depending on the number of how many you have initially, sometimes it sort of they all go extinct, or sometimes it sort of grows, right? If I understood this correctly.
Right. So it seems that there is if you start with very small numbers and you start adding them, it's very hard to get going. It seems to be an exponentially rare event to get the big enough number that you can go up. There's a critical number roughly above which it's very likely to go up. And then the bigger you get, the less likely you are to come crashing down again. So there's sort of a nucleation problem. We don't know how special that is to the particular model and things. We understand why there are some relatively large numbers coming in here because of the big ranges over which things can fluctuate. But we don't know whether there are sort of simple situations where you can start with small numbers and diversify. That we don't know yet.
And do you understand this threshold? What sets this threshold? That's sort of interesting, right?
No, I think it probably goes as the square of the logarithm of the migration rate in
but in your matrix elements, the variance does not scale with n somehow, right? So as you add more species, you go over the May limit in some way, right?
We're always over the May limit.
Yeah, so that's very interesting, right? Yeah.
So we from the beginning we say we want to throw away the people can still see my screen.
Yes, I can.
Yes. Okay. So this was in this phase diagram I had here. So really, the niche term has to be of order the square root of the number, right? So what people normally do is they scale it in such a way that they scale the interaction between them down by 1 over square root of k. Then it looks like things order 1, but that's a very unnatural thing to do when you've got strains within the same species. So we're basically, we would argue that we're sort of always down near this end, so that we focus on that. If you come up here, so you increase the number of strains, and you come down through there, because this convenient scalings for doing the math. But then there's a question: what are reasonable scalings as far as even biologically motivated in a very, very loose sense.
Okay, thanks.
Thank you, Tobias. Let me ask you just one final question. Do you think it's feasible to test this experimentally to track the number of species and to see if they follow some kind of chaotic dynamics in some control or non-control environment? That'd be really nice.
It's difficult. What we're hoping, so Prochlorococcus, which has an advantage, it's the dominant producer in all the tropical oceans, which are very nutrient poor. So there are not many others. So it's a very substantial fraction of all the microorganisms in those oceans. And there have been tremendous amounts of samples collected and sequencing things done by Penny Chisholm's group, who actually discovered them ages ago. So tracking things with time and things there and looking at genetic differences, one can do. The problem is everything's getting mixed around, and you can try to do it in the Lagrangian frame, which they sort of tried to do. Doing things in the lab, I think the bacteriophage, there are species now which are known that the phages have broad specificity. They don't just attack one strain, they attack a whole range of strains. And so trying to do things where you have phages which attack a whole range of strains with different strengths, and then looking at how that behaves in the lab, including under evolution, I think is a viable experiment. As far as I know, nobody has tried to do something like that. But you can start with a pool where you can measure the interaction between the bacteria and phage, a whole bunch of them to begin with, and then put them in together and ask about what happens. So I don't know much at this point. It's too early to really say about sort of concrete predictions and things. And really the goal is to sort of make a scenario. Because basically I would say there are essentially no scenarios for explaining why. This is the thing I've been involved with my colleague Remarkably. You take a little sample from the top layer of the microbial mat in a hot spring in Yellowstone. And you look at one species in there, again, it's the dominant reducer. Every cell you pull out is different. Some of them diverged a million years ago. Some of them diverged 100 years ago. They're all different. They're all mixing together. They're all competing. They're moving around somewhat and things. What on earth could make them so diverse? And there's a lot of things you can infer from the genetic information that that there has to have been a lot of selection and things going on. They exchange genes a lot. They have a huge amount of recombination between them. So in some ways, it may be a problem of trying to understand the diversity of bags of genes, which is more like human diversity than the diversity of like asexual genomes. The difficulty with microbial biology, well, I mean humans to some extent also, is you can do sequencing out the wazoo. any measurement you can turn into a sequencing measurement is you do it. But the problem is you still have very little idea of the connection between the sequence and the phenotype, the organism's properties. And that's true even in the immune system, where the relevant sequence is only sort of 100 bases. And we still don't know, when you understanding what's going on in the evolution of the immune system in a person is still limited by the fact that we don't know the connection between the DNA sequencing and the. So what one needs is you need predictions as to how they will show up in genetic diversity. And if you can get in a situation where you can measure dynamics, then one would like that. But at least one should be able to get predictions for properties of the static diversity and ask what things will at least rule out scenarios like neutral ecology, and try to, if one has enough predictions, to be able to to rule out some other things and distinguish between scenarios. But that's a very early stage.
Okay, thank you very, very much. So I think we can close the session today. As I said before, I really enjoyed both of the talks. It was great. So I hope everybody also shared my enthusiasm. Thank you again, the speakers. Thank you, Julia. Thank you, Daniel. It was a real pleasure to have you here. And,
thank you very much. I wish I was actually there, but hopefully we'll
next time. One
of these years.
And see you tomorrow in the next session of the Granada Seminar.
Thank you, everyone, for the very stimulating questions.
Thank you. So see you tomorrow. Bye-bye.